Now we'll discuss the life cycle of Entamoeba histolytica. So first of all, as I mentioned earlier, the infective stage is trophozoite. So the trophozoite is the stage in which it enters into the human. So you draw a trophozoite here. This is the nucleus. Here you show RBC. This is active amoeba. Okay, it has entered into blood of large intestine. So now what you'll see is this will get converted into pre-cystic stage, the one we draw earlier. So this is it. This is the pre-cystic stage. This is nucleus. Then it will get converted into what I drew earlier, that is Uni Nucleate Stage. You have to draw chromoid bars here and glycogen vacuoles, nucleus. So this is Uni Nucleate Stage. Now what you'll see is it will go into the fecus and get converted into binucleate stage. Then it will get converted into what we know is having 4 nucleus, the quadrinucleate stage. Then it will undergo excistation, and the cyst wall will be removed. Excistation. This will lead to the progeny amoebas being released. And then they'll go into the blood and get transmitted. Now what I want to clear out here is from this stage till uninucleate, this change occurs in large intestine. Till here it occurs in feces. Here it occurs in small intestine. Okay. Now what I want you to know is, as we saw in this life cycle, there is just one host. Okay, so single host, which is man, obviously. Then how is the infection acquired? Okay, mode of infection, it is through contaminated food and water with which stage? Quadrinucleate stage. With quadrinucleate stage. It is also transmitted through anal or oral sexual practices in male homosexuals. Now there arises a question that why is the cyst wall still intact in intestine? Because it is resistant to the action of trypsin. Now what happens is, during the growth they form a flask shaped ulcer. So some of the amoebae, they enter into the blood and go through the mucus in feces and get excreted out. So this is amoebic dysentery. But in some, they in liver they will multiply, and then they'll lead to the occurrence of amoebic hepatitis or amoebic liver disease. This I'll discuss in pathogenesis. Another point that you need to remember is that it has an incubation period of 4 to 5 days. we'll discuss about the pathogenesis in under two headings, that is the primary and the secondary lesions, and then we'll discuss the lab diagnosis.